All right, so I'm just going to quickly review the things we talked about in the last one, and then I'll pick up with some new stuff today. Fungi supposedly evolved from unicellular protists. They sort of look a little bit like plants. Obviously, most of them aren't green, though. They don't do photosynthesis. But genetically or phylogenetically, evolutionarily, they're a little bit more related to animals. They're heterotrophs. And the way they digest their food is extracellular digestion. That means they basically spit out enzymes under their food, digest that outside their body, and then suck in the juice. That's why things that are moldy get all soft and gooey, because they're digesting that stuff outside their body and then sucking it in. They do have cell walls, but they're made out of a little different sugar than the sugar we find in plant or bacteria cell walls. It's called chitin. Most of them are multicellular. Now, there's some exceptions, like yeast, which are one-cell things. They're all eukaryotic. They have a true enclosed nucleus. They're all heterotrophic, and sometimes we call them saprophytes. That means they sort of look like plants, but they feed on what? Dead stuff. They're decomposers. Most of their life, they're haploid. They have one set of chromosomes. That lets them do asexual reproduction, do it really, really fast, so that they're really good at competing for things, for resources. But they do have a sexual stage, and during that stage, part of the time, they're diploid, two sets of chromosomes. And obviously, the sexual reproduction is about getting some genetic variation. Most fungi grow the best in places that are warm and moist. That's where you're going to find fungi for the most part. Now, we already talked about saprobes and saprophytes, how that they feed on dead things. They're made of these little string-like filaments called hyphae. That's what a fungus is made out of. And these intertwine and make a big mass of things called the mycelium. Anytime, again, you see M, Y, and C in this class, you should think fungus. It's a fungus body, is what we call that. Some of these hyphae are septated. What last time did we say a septum was? A septum. What's a septum?
Is that something like?
Yeah, it's like a wall between your nostrils, between the sides of your heart. So most of the time, the fungi hyphae look like this, with walls in between each cell. But there are a few where they have these hyphae that keep dividing, but they don't make any walls. So it's like one long cell with lots of what those dots represent. Nuclei. Those are called cenocytic. If you see that word fruiting body, realize that's the reproductive organ of a spore, of a fungus rather. And if you see the word conidia, realize those are spores that were created asexually, not through sex at all. We talked about how they reproduce. Let's go to this picture to deal with that. First of all, lots of times they can reproduce asexually, where the mycelium grows a spore-producing body, a fruiting body, and then that makes spores through what process? What would be the asexual part of cell division? By what? Asexual. No. Mitosis. Okay. Binary fission is a bacteria thing. Don't confuse that with anything else. That's a prokaryotic thing. Here they produce spores by mitosis, and then those just grow into another fungus. That's asexual.
Asexual, they just reproduce spores by
mitosis. But the sexual part's a little bit more complicated. In that, two different fungi grow together. Their hyphae bump into each other. Those little filaments bump into each other. They fuse. And we get something called plasmogamy. This means joining. And joining of what? What's the plasma referred to? Cytoplasm. Joining of the cytoplasm. The cytoplasm is joined together. At that point, you've got one cell with two nuclei. That's why it's called dikaryotic, two nuclei. Pretty soon after that, you get the joining of the nuclei. So again, joining of nuclei. And that gives you a cell that's diploid. Diploid meaning it's got two sets of what? Two sets of chromosomes. Just like you. Yep.
So what is the plasma?
The joining of the cytoplasm.
Okay.
Right. This stage of karyogamy is sort of like fertilization in a person. Sperm plus egg. Haploid plus haploid gives you diploid. Once that happens, the cell starts to undergo meiosis. Meiosis takes diploids and splits them and makes what? Haploids. So that seems kind of backwards. That you started with haploid, you made diploid, and now you're going back to haploid. But meiosis does something else. What else happens during meiosis that's important here? It takes diploid and makes haploid, but what else does it do to the chromosomes while it's dividing them? It mixes them. It's like shuffling the deck. It's shuffling the genetic card deck. So this right here, this meiosis step, that's where the genetic variation comes from. It grows after meiosis or during meiosis, it grows a spore-producing structure, a fruiting body. Out come the haploid spores. Those grow into a new fungus, and we start all over again.
I miss karyogamy.
It's the fusion of the nuclei. G-A-M-Y is fusion, karyo is nucleus. All right, now let's backtrack a little bit. Last time we started talking about the different fungus groups, and we talked about this first one, not as much as we're going to say about it. First group is called Basidiomycetes, and that means clump fungus. Before we go into this, the way they're divided into groups, the fungi, is based on how they sexually reproduce. These have a club-shaped reproductive structure called a basidium or basidia. These are probably the most common kinds of fungus, at least the ones you're most familiar with, like mushrooms, puffballs. And these things, mycorrhiza, which we'll talk more about in a minute, they're fungi found on roots of plants. That's a good thing for both. But the main role of these guys in the environment is they're decomposers, and when they're decomposing things, they're recycling those minerals. They're putting them back into the soil. Mineral recyclers. In terms of the way they reproduce sexually, it's just what we just talked about. Two haploid hyphae cells fuse, plasmogamy. Pretty soon after that, the two nuclei fuse, karyogamy. After that, we get meiosis. Meiosis makes haploid spores, and then those grow, germinate, and we start all over. Got some pictures to show you. There's some puffballs. That's corn smut right up here. Looks like a big tumor growing on the corn. There's some mushrooms. Last time we also briefly mentioned these guys, ascomycetes. Again, there's that MYC, fungus root. These are the sac fungi. Their reproductive structure is called the ascus. This is where they make spores. There's several different ones. Some of these you've heard of, some of them maybe not. Truffles are sort of like mushrooms that grow into the ground. Penicillium obviously makes penicillin. The different kinds of yeast, like Candida, for example, is a type of yeast. Saccharomyces is a type of yeast. Sordaria is a fungus we're going to use in class here in a couple of weeks during one of our meiosis labs. Also lichens, which we'll see in a second on our PowerPoint.
So what is yeast infection stuff from
These things. These are Candida is the one that causes yeast infection. There's some truffles. Those don't look very appetizing to me. Those are some yeast. This is just showing you that the way the sexual reproduction in these guys works is basically the same as what we described. You've got two hyphae, two long threads that eventually bump into each other and join. When they join, that's called plasmogamy. that leaves cells that have 2 nuclei in each cell. You can see those cells in there have 2 nuclei. Those are called dikaryotic. 2 nuclei. Eventually, those nuclei fuse together. And we can see that up in here. That gives us karyogamy. That gives us a diploid nucleus. That cell then undergoes meiosis. Now something you should see, meiosis takes one diploid cell and creates how many cells from that? Nope. One diploid, 2N means diploid, to what? How many do you see here, guys? One became 4. Okay? And those are haploid or N's. Now notice, right after meiosis, though, this became 8. How did we go from 4 to 8? In fungi, after meiosis, what happens right after it? Mitosis. So you get 1 becomes 4, and then mitosis just copies each one of those and doubles it. That's why we've got 8 in each one of these sacs. These are the ascus or asca sacs. That's why they're called sac fungi. And they're always pretty much going to have 8 spores inside of them. All right, another group are called zygomycetes. Sometimes these are called terrestrial fungi. Normally grow on land growing fungi. These are common things too, like bread mold. That's stuff that grows on oranges, fruits.
Greenish.
What now?
Greenish.
Yeah, greenish, bluish kind of stuff. Turns out there's several of these that are disease causing. Some mycorrhiza, again, the fungus that grow on plant roots. And also penicillium, they're debating on which group it ought to be in. Notice it was listed on the last one. It's listed on this one.
Does that have characteristics from both sides?
Yeah, what determines what goes where? Yeah, but in general, what determines what group something goes in?
Characteristics.
Phylogenetic.
Phylogenetics. So there's some debate about which group it should go into. There's some bread mold. Not going to say a whole lot about that. Notice here's their sexual reproductive cycle. Actually, here's the asexual cycle down here. These are fruiting bodies. And notice they make spores by mitosis. Those are dispersed and germinate and grow into a hyphae, and then we start over. Up here's the sexual part. 2 mycelia, 2 hyphae rather, fuse. We get plasmogamy. After that, the nuclei of those cells fuse. We get karyogamy. That goes through meiosis, creates haploid spores. Those get dispersed, germinate, start over again. So they're all reproducing the same basic way. 3 steps. Well, actually 4 steps. Fusion of hyphae, plasmogamy, karyogamy, and what's last? What's last? Meiosis. With the spores. All right, another group are called the deuteromycota. Again, MYC. Notice those letters, fungus. These are sort of like the protists in that taxonomic holding station. What that means, what did we do? What did the guy in the video call the kingdom protista, what it used to be kingdom? He said it was sort of like the junk drawer, right? They put things in there that they don't really know where they go. That's sort of what deuteromycota is. Here, scientists haven't figured out how these things sexually reproduce. Maybe they don't. But so far, things in this group, they can't figure out how they have sex. Candida, that yeast that causes yeast infections, penicillium, they both used to be in this group. But eventually, scientists figured out how they were reproducing. And now they moved them to the appropriate group after they did that. The one thing that all the fungi in this group have, scientists don't know how they sexually reproduce.